doesn't follow me, right? Good, I'll stand over here. Okay. So, we ready for absorption? All right. This is just the first part of absorption. We'll just talk a little bit about sort of the theoretical underpinnings of it, and then talk a little bit about those types of materials that are absorbing. And then we'll follow up tomorrow on measurement and thinking theoretically about how we make an accurate measurement for absorption, and then the reality of how we actually measure absorption and what those assumptions are. So, okay, so we'll talk a little bit about the electromagnetic spectrum, what happens when we have absorption on molecular scales, and the absorbers. Okay, so if we have a dipole, a charged particle, they will create electric fields. And it will look something like this. There'll be an oscillation where the vectors are indicating the magnitude of the energy, and that will oscillate. Now, when this charged particle might move, it's going to create a magnetic field. And you'll get a magnetic field. In this case, it's like in this barely visible blue field vectors. And you can think about this as these the energy is propagating with these fields. And if you remember the right-hand rule, that if you take your fingers and you point them in the direction of the electric field, and you curve them into the magnetic field, you get the direction of propagation. Okay. And so the frequencies that we're thinking about are described by the electromagnetic spectrum. And so in this diagram, you've probably seen a million times, you can see that we've got from gamma rays to radio waves. Visible light is actually a really tiny part of this spectrum, and that we have increasing energy as you go to shorter wavelengths, and we have increasing wavelength as you go to lower energy. So, what did I say? So, when say a molecule absorbs electromagnetic energy, it is going to really depend upon which part of the spectrum that you're in. And that has to do with the fact that we have this relationship between the energy and the wavelength. So if you're in the visible part of the spectrum, there's going to be a certain amount of energy associated with that, and that's going to do a couple things to the molecule. If you're in the ultraviolet or you're in the longer wavelengths, different things can happen to that molecule. In this relationship, the energy is proportional to Planck's constant, which we saw in Planck's equation, the speed of light in a vacuum, and the wavelength. So let's look at what's going to happen to molecules as they absorb light. So here's our electromagnetic spectrum. I'd never really thought about this as being like the extreme UV, but there is actually a part of the spectrum that's called the extreme UV, which is like really short. So when molecules absorb energy in the microwave region, what that does is it sets up a rotation. This is the spin. And that's why microwaves heat up food, right? Kind of clever. If they move to absorb in the infrared, say for example a water molecule, that is setting up a vibrational energy in that molecule. So it absorbs a certain amount of energy and it just starts to vibrate. Okay. If we get into this region here in the visible and ultraviolet, what happens to the molecule is you can actually transition electrons to other orbitals. And if you get to X-rays, you can actually ionize those electrons, and over into the gamma rays, change the nuclear configuration. So these are the bad areas where you want to stay from that. So we know that if we have molecules that are absorbing visible light, we're going to see changes in this electron state. And so let's think about looking at this energy diagram. And this represents the ground state, and these represent the higher electronic states. And so we're going to build a chlorophyll absorption spectrum. Okay? So if we have this molecule and it's absorbing amount of energy to move the electron to the higher orbital, we could see this as this arrow here, from one electronic state to the next electronic state. Okay? Chlorophyll actually has the potential for two different electronic states. Turns out one is in the blue, the energy equivalent of a blue of blue photon, or the energy equivalent of the red. Right? And red has less energy than blue. Right? And so we could plot this as absorption versus wavelength. So we would have a quantized amount of energy associated with this wavelength here, and a quantized amount of energy associated with this wavelength here. And nothing here, and nothing here, or here, or here, or here, but only here, right? That's the electronic state. And this is what's happening on a molecular level. And it turns out that, of course, molecules can only absorb energy of that quantized amount, right? They can't absorb a little bit more. So now we have our peaks. And for chlorophyll, we have a peak in the blue, about 440, 436, and around 676. But you say to yourself, that doesn't look like the absorption spectrum that I'm used to seeing, right? And so we also have quantized vibrational states. So each of those orbitals, those electronic orbitals, is associated with a series of higher excited states. And then, oh, there's my diagram. Okay, so while the electrons are moving orbitals, the bonds are also vibrating. And so within each one of these electronic states, there are these vibrational levels. And so you can imagine that if the molecule was in this higher vibrational level, now it can absorb amount of energy to this electronic state, or this one, or this one, or this one. I'm sorry, this one, or these here. This one could absorb up to this electronic state, or within these vibrational levels. And so if you remember that the length of this represents a quantized amount of energy, and each quantized amount of energy is associated with a specific wavelength by hc over lambda, that allows you to see some absorption in some wavelengths beyond that main peak, which is the electronic state. Okay? So now you have absorption here, but not here, here, but not here, here, but not here, etc. Okay? So then, and the difference in this and the energy of these vibrational states, remember, that amount of energy is associated with a certain, with a change in wavelength that's equivalent to thermal infrared, right? That's the vibrational amount of energy. And so it's a very small difference between here. So now we can go to the quantized rotational states. And again, this is involved with this process in the molecule. And these are the little dashed lines which represent rotational states within each vibrational state for each electronic state. And that's how you begin to fill in the potential for absorption of energy across a spectrum. The major peaks are electronic states, where electrons move to another orbital. About that, we have vibrational states of the molecule. So at the same time, while it's moving to another orbital, there's a vibrational state associated with that molecule, unless it's at absolute zero. And then there will be rotational states. And so that's how you build an absorption spectrum. So when you think about molecules and you think about absorption spectra, and you see peaks that have one, or absorption that has one peak, you can think to yourself one electronic state. And if it has a very narrow peak, you can think relatively narrow vibrational and rotational states, mostly vibrational. So you can begin to think a little bit about molecules once you see their absorption spectra, and particularly the location of those peaks. Right? If you have peaks in the UV, you know that those electronic states are very far apart, because that's a very high energy in the ultraviolet, right? So things like proteins, which absorb in the UV and have peak absorption in the UV, have a very big energy difference in their electronic states. Okay. Okay. So chlorophyll has these two electronic states. And here's the chlorophyll molecule. And I know Ivana is going to show you the chlorophyll molecule again. You'll get to see or hear about it a couple of times. It's a really interesting molecule. It's got this chlorine ring structure here. It's got a magnesium in the center. And then this chain of carbon chain called a phytol tail. This is actually what holds it into the the membrane in the chloroplast. And then this face sort of sits up, available to absorb light. And it's this structure here that's responsible for absorption. Okay. We'll talk about fluorescence, I think, next week. So I'm not going to talk about that right now. Okay, so just to show you how this molecular structure can impact the absorption spectrum, there's the molecule again. There's the blue peak and the red peak. So those are, in this case, these are some other vibrational states. Now, if you take that molecule and you put it into acid, what happens to it is that you lose the center magnesium. Okay, how many people have ever done extractive chlorophyll technique? Where you filter water? Oh my gosh, a couple people. Right? You filter water, you put it on a filter pad, you put the filter pad in acetone, and then you measure it on a fluorometer, and then you add a little acid, and then you measure it again. This is what you're doing. Okay? You're pulling the magnesium out of that ring structure. That's not the one I want. I wanted this one first. Okay. If instead you were sloppy and you let your sample heat up, you can lose the phytol tail, right? So here we don't have the tail. And that would bring you to a pigment called chlorophyllide as opposed to phaophytin. And then if you were like, oh, and now you make your measurement by mistake and you measured it and then added acid, then you would remove the magnesium. So in this method, one of the things we're going to talk about is once you have it in extract, don't let it heat up because you want to maintain this structure in the estimation of chlorophyll, and you'll be able to measure chlorophyll and phaophytin. And the reason why phaophytin is important is because what could you imagine in the natural environment might change chlorophyll to phaophytin? If you have phytoplankton that have chlorophyll pigments, how could you ever acidify them in nature if something eats them, right? And so zooplankton grazing on phytoplankton will naturally remove the magnesium. And so if you measure phaophytin in the environment, you give an estimate of maybe some grazing pressure. So you'd want to measure how much chlorophyll is in the water and how much phaophytin is in the water. But if you are sloppy and you let your sample heat up, you're going to end up with chlorophyllide and phaophorbyde, which are very different. And I don't think that this fluoresces very well, and so your sample will be biased. Okay, so the interesting thing is that if you remove the phytol tail, it doesn't change the absorption properties too much. There's some changes down here. There's also this other absorption around 460 by moving the tail. So removing the tail doesn't change absorption that much. Removing the magnesium, you lose this peak, but not this one. There's that peak. I can't find one person who actually made all of these measurements at once. But you lose this blue peak of absorption, which is what happens in the formation of phaophytin as well. You keep this peak, but you lose this one. And it also changes, it shifts a little bit the red peak, and the fluorescence changes. So while you're making these measurements of absorption and fluorescence by molecules, you need to be thinking about what is happening to the shape of the molecule, how that's changing its absorption spectrum, how it's changing the location of those peaks, because it's changing the bond structure. And it's the bond structure that determines where the electronic state is and what amount of energy is being absorbed. And it's this sort of outer ring of double bonds that is responsible for those electronic states of absorption. Okay? Any questions on that? Okay. All right. So let's look at the major absorbers and see what we can learn about them. So if you went out, I think this is probably taken from my PhD thesis. I don't know, looks like the font looks like it. If you go out and you measure absorption in the open ocean or a coastal environment or an estuarine environment, you can get these very three different sort of characteristic absorption spectra. And again, we're looking at the absorption coefficient from blue to red. So my question is, what do they all have in common? Yeah, they're pretty similar over here, right? And then they're really, so they all have strong red absorption, and then they differ in here, right? And it's pretty obvious because you know that as you go from the open ocean into coastal environments, you've got a lot more stuff in the water, a lot more material, organic and inorganic, that can contribute to absorption. But all of those locations have water, and water is responsible for this absorption, right? Okay, so the cool thing is that that absorption is a conservative property. And that means that if you could measure the absorption by water, and if you could go out and you could measure the absorption by all the different dissolved compounds that we have, all the compounds that are part of CDOM, color dissolved organic matter, and if we could measure the absorption by all the different particles, we could add them up. You could get the individual absorption spectra of each particle and add up all the particles. Or you could figure out a way to just measure the absorption by all the particles at once. But because it's conservative, we can add it together and get that total, which is really helpful. And then the other thing that's really nice about absorption is that it's proportional to the concentration. And we'll do some experiments tomorrow, sort of looking into the details of Beer's law. But it basically says that if I have a compound, say chlorophyll, and I've measured its absorption spectrum, and I've divided that spectrum by the concentration, or come up with the absorption by chlorophyll for, say, 1 milligram of chlorophyll, right? The mass-specific absorption. And we often call that A star of chlorophyll. And it will have these really intuitive units of meters squared per milligram. But that's because absorption has units of per meter. And so once we've established that we know what the absorption spectrum is for a chlorophyll molecule, or for a milligram, or for a mole, however you decide to do the absorption, then if you go out and you measure the concentration of chlorophyll in the water and say it's 7 milligrams, you can just multiply that by this spectrum that you've had to measure once. And you can achieve what the chlorophyll absorption spectrum will be. And so all you have to do is measure this. And so this holds for every constituent. We can be for CDOM, it can be for phytoplankton, it can be for non-algal particles, it can be for inorganic particles. Any of the absorption components, if you can define its mass-specific absorption spectrum, all you need to do is go out and measure the concentrations of what you're interested in. So it's really powerful. So thank you, Beer's law, for that. Well, thank you, physics, for that. Okay, so we tend to divide absorption into these components. We say that the total absorption is equal to the contribution by water, the contribution by color dissolved organic matter, phytoplankton, and non-algal particles. And so these are some examples of what the molecular structure of CDOM looks like. We don't really know what it is. There's people spending careers trying to figure out what CDOM is actually comprised of. We've got some phytoplankton here. This turns out to be an Alexandrium fundiense, which is common in these waters and is responsible for the production of saxitoxin, which causes paralytic shellfish poisoning. Fun fact. And here's a bunch of zooplankton and bacteria and junk, living and dead. They show up in this non-algal particle component. And then here's some nice inorganic sediment that shows up in this also because it's not a phytoplankton. Okay. But when we figure out how to make these measurements operationally, there's a little bit more complication that goes on in there, which we'll talk about tomorrow. Okay. So we group these because these have different roles in the community, but we really group them because it's the only way we can separate absorption when we're making the measurements. We can filter out the CDOM. We can separate phytoplankton, because really the part about phytoplankton that's interesting in their absorption is their pigments, and we can separate that out. And then we're just left with all this other stuff here. So we divide it because we can. But then interestingly, we divide it because they have similar absorption characteristics. CDOM, while there's a variability in CDOM absorption depending upon its composition, it looks more like itself than it looks like anything else. Non-algal particles, living and dead, inorganic, organic, they look more like each other than they look like phytoplankton. So, and they're particles. So we separate them. They are sort of more similar to each other than they are to any of the other components. So the fact that we can separate these and the fact that it is useful optically is great and good for us. Thank you, Mother Nature. Okay. So let's start with the components. This is an absorption spectrum by water. Through the ultraviolet all the way to the far infrared. And this is a log scale. So water has tremendous variability in its capability for absorption. And where does it not absorb? In the visible. So that's nice for us. So we have this little tiny window in here in the visible where water does not absorb. But basically, as soon as you get into the near infrared, it's strongly absorbing, so it makes it a greenhouse gas in the atmosphere. And it's very, very absorbing of the UV, which is why you don't get sunburned if you're scuba diving without a scuba suit on. So snorkeling deep and holding your breath for a long time. Okay. And you can see all these vibrational states in the absorption spectra of water. Okay, but we're going to focus in on the visible. So if we have it on a linear scale, the absorption looks something like this. If we have it on a log scale, you can see that there's some minimum absorption right around 400. So it's very blue, which is why, of course, the open ocean is blue. Now, a bunch of years ago, Scott Pagaw looked at absorption by a bunch of different labs and plotted everybody's data in this really old graph, but plotted everybody's data, everyone who said, we've got the absorption by pure water. But you can see on this log plot that they did a really good job out here because the signal's strong. It's easy to measure. When you start getting into where the absorption is low, it's really tough to make the measurement. And it's really tough to get clean water. And so a lot of this is methodological, right? You got to get clean water, which is tough to get, and you got to have a good instrument. So we also have some natural variations. So if a single lab made some measurements of pure water, it looks pretty repeatable until you get to this area. And it turns out that it matters what temperature the water is. And this is actually a natural variability. So if you're in the Arctic, the absorption is lower than if you're in the tropics. Okay. And the reason is that when water molecules, was it Emmanuel? He said he wanted to be a water. You wanted to be water? So do you want to be the molecule? Do you want to be the cluster? Do you want to be the fluid in a Bernoulli, a turbulent or laminar flow? Which can be turbulent or laminar in flow, so multiple molecules. Definitely laminar flow, sometimes turbulent. So, I'm sorry. So the reason is that when water molecules are next to each other, it's a polar molecule, right? Like the oxygen has a net plus or negative, and the hydrogens have a net plus, and so they get these hydrogen bonds between them. And if you cool it way down, of course, they lock into a lattice structure of ice of 6 molecules. But if it's cold but not frozen, and there's not a lot of kinetic energy or vibrational energy, the molecules can pack in, and they're not moving very much. And so you get lower vibrational energy in here, and you get a change in the absorption. Whereas if it's really warm and the molecules are really vibrating, it changes the molecular structure of those molecules next to each other in the hydrogen bonds, right? So this is a natural source of variability, and we're going to see it in the lab too. And it turns out what is a natural variability absorption ends up being a pain in the neck methodologically, okay? Okay. Oh, and yeah, there's the number of molecules per cluster is really high at low temperatures, and the number of molecules is low at high temperature. So if you look up clusters on the internet, it will talk about microclusters of water molecules. And that if you have water molecules that only have a couple of molecules in there that are clustered versus large clusters, that it has different nutritional value. You don't hydrate as well with macroclusters. You need microcluster water. You need ionized water with microclusters to properly hydrate. Or you could just drink more water. Anyway, there's no truth to the matter, but it's interesting. If we pick a wavelength here, say 740, we can actually look at the difference in the absorption as a function of temperature. And so we'll be actually doing that in the lab. That's important to know. It only shows up in these areas of the vibrational, quantized vibrational states. And the biggest one is out here in the red. Okay, similarly with salinity, when you add salt to water, it changes the structure, and you get the negative polar parts of water wrapping around like positive chloride or positive sodium ions, and you get the positive parts of the water molecule wrapping around the negative chloride. And so there's definitely changes in molecular structure. And so Pega and his colleagues found that salinity causes, as you increase the salinity, you get a decrease in the actually this is beam attenuation, but it's also because of absorption too. And then Sullivan found that you get an increase if you add salt. Both pretty good scientists. So you have to actually go and look at the spectrum to find that sometimes it gets higher and sometimes it gets lower. So it really matters what wavelengths you're looking at, right? What the effect of salt is going to do to the absorption spectrum. So we'll look at the effects of salt as well. Okay. If we look at dissolved inorganic matter, like the salts, you tend to see the effect of them absorbing mostly in the UV. And so, for example, here's bromide. It's a very strong absorption. And these are, this is on this scale. Very strong absorption, right around 200 nanometers. And if you're measuring absorption of samples in a spectrophotometer that goes to the UV, you'll see this really strong increase in bromide. But we can also see things like nitrate, hydrogen sulfide, a variety of different molecules. But they all have this really strong absorption in UV. And so this is actually the basis for the detection of nitrate. You can use an ultraviolet spectrophotometer to measure nitrate concentrations in the water, which saves you a lot of chemistry if you calibrate it. So it's all based on absorption. Color dissolved organic matter. So we take a water sample, we filter it, and we put the filtrate in a cuvette. Save this for later, the filter, which has all the particles on it. And then you measure the spectrum, and you find that you get this exponentially decaying absorption in the visible. So we haven't quite gotten to its peak, right? There's going to be a peak somewhere out here. We don't usually get to it. Here's some examples from some inland waters. They tend to have really, really high absorption in inland waters. This is from Australia, John Kirk's book. And this is when you can get this reddish water. You'll see this type of red water in many, many of the rivers around here and many of the lakes. So if you're interested in CDOM, you have come to the right state. Okay. Sometimes you can put your hand in it and not even see your fingers. There's so much CDOM in the water coming from all of the wetlands around and all the marshes and the wave, right? Okay. So what people notice is that you can actually fit a curve to this, right? It's an exponential curve. And so it fits very nicely this model, which says that if you could just measure CDOM absorption at one wavelength, say you had a one wavelength absorption meter, you could predict the entire spectrum if you knew what the slope of that was, right? So knowing something about that slope, how the exponential slope, how steep is it? Is it a flat slope or is it a steep slope? Can tell you. You'd be able to actually not even have to measure the whole spectrum. So being able to have an analytic curve to absorption is really quite helpful. So Ken Carter did a lot of work in the Mississippi, and he made measurements in the loop current in the Mississippi River, and he had absorption spectra that looked much like this, but then he plotted them on a log curve, or I'm sorry, a log transform axis. And of course, if you have an exponential, it ends up as a line on a log. And he found that there were really significant changes in the slope of that absorption curve. And so he got some samples of pure fulvic acid and pure humic acid and actually found that you can model the change in the slope, how steep that exponential is, based upon if the material is more fulvic, which is sort of more like tea, and it's refractory material. It's probably all been eaten down by bacteria to the part of the organic molecule that there's just not much nutritional value left, compared to the humic material, which is the fresh organic matter that would come off soils or come out of a phytoplankton bloom, right? So that's change in slope. Excuse me, you just spit it all over everybody. This change in slope is actually really important for discerning what's in the water and where its source might be coming from. Okay. And so the slope of this line would be this value here. Okay. And so this is just the modeling that he did. If you did pure humic and pure fulvic, he found that the slope was 0.01 versus 0.02, which sounds like, really? Is that really that important? But it turns out you can really resolve this. You can resolve this down to 3 significant digits. So I had a student who went to the equatorial Pacific and went along the equator and did profiles. And everywhere there's a dot, that's a water sample. And she collected whole water and filtered it at 0.2 microns, because that was the technical filter size for CDOM. But we also did a lot of work with glass fiber filters. So she also did some filtering with glass fiber filters, which have 0.7 microns. So there's not a tremendous difference between 0.2 and 0.7. But if you difference it, you can get this interval of 0.2 to 0.7, which is colloidal, a colloidal fraction of organic matter. And what she found was that the part that was really small tended to be located down deep, except in this upwelling zone where it was actually being upwelled to the surface. And the other material tended to be at the surface, this colloidal part. And then when she looked at the absorption spectra for those two size fractions of dissolved matter, she found that this was steeper than this one, which suggested that this was more refractory material. And so we're at the equator where we're having upwelling of deep water that is really refractory, and turns out is really old. Has been there for a long time. It's been eaten down by bacteria, and you're sort of left with this residual old carbon. Compared to this material, which was associated with phytoplankton, which was probably freshly, released into the water because of grazing or leaking. And so by being able to measure spectral slope, it tells you a lot about the material. And then by looking at the distribution, it tells you a lot about the oceanography. And so something so simple as fitting an exponential curve to CDOM can be really instructive and provide you with tons of information. Okay. So I was very interested in CDOM when I was a student, and I did not have an opportunity to go on a lot of cruises, but I had the opportunity to digitize everybody else's data. So I did, and I digitized data. Any published spectrum I could find, I digitized it, and then I computed the slope. And I found that I could calculate a mean slope, and then about 10 years later, no, a little more than that, 15 years later, Marcel Babin did a big giant cruise around the European waters and measured 346 spectra and calculated slopes and did a histogram of the slopes. And what you find is that the slope of CDOM absorption is relatively narrow. And the values for the global ocean are not significantly different from the values from all of these measurements in the European waters, which suggests that it's when you see these extremes that you can get some information. It makes a difference if it's 0.01, 0.06, or 0.02. So the difference between these here is composition. But there's a relatively robust slope that you can calculate. Okay. Any questions on CDOM? So fascinating. Okay. So in your sample, which you will do this week, you will also then collect this filter. And so that filter has all the particles on it, right? And so you're going to scan it, and you'll get the absorption of particles. So if you happen to have a culture, your absorption might look like that black line there. 2 peaks of chlorophyll, some other stuff in there. If you were out in the bay, you might see those 2 peaks, but you might see some other sort of background stuff in here. It's not quite as clean. So then what Kashino did is thinking, well, pigments can be extracted with methanol. He took this filter, put it back in the filter cup, put methanol on it, and extracted all the pigments out. So the interesting thing about doing that is that it leaves all the other particulate matter on the filter, including the phytoplankton cells. It just takes their pigments out. And if you look at it under a microscope, which I've done, the cells are perfectly intact. You can see all their organelles. And you can see all the other detrital things in there too. So it's almost as if you just waved a wand and got rid of the pigments. It's actually a really simple but really powerful technique. And then you scan it again. And since you have all the other organic and inorganic living and dead particles on there, that's non-algal particle absorption, right? And so if you do it for a healthy culture, that scan should be pretty low. It can tell you if your culture is healthy. If you did it in the ocean, you might get something that's a little bit larger, right? And it's got, what sort of shape is that? Exponential again. Is that a surprise that organic matter in a particulate form and organic matter in a dissolved form have a similar absorption? It was not a surprise to me. It's not a surprise to Eric. Because there's a continuum of size, right? There's a continuum of living organic matter to dead organic matter to colloidal organic matter to dissolved organic matter. And so we would expect that the molecules that are responsible for the absorption are going to be the same across that material. The other cool thing about this is that if you subtract that non-algal particle absorption from the original particulate absorption, you obtain the absorption spectrum of the pigments as they were in the cell. It's very different than extracting the pigments out into a solution and measuring those, where the pigments are all flying all over in their solution. When they're packed in the cell and they're rooted into membranes and they're attached to each other, their absorption spectra is very different because the molecular structure of the pigment is different when it's attached to a membrane. And if you want to know what a phytoplankton really absorbs like, you need to know what its absorption is intact or in vivo. And this is how you get that. Because this first scan has them in vivo. in the cell, packaged up. This second scan, they're gone. And the difference is the in vivo absorption spectrum. Very simple, very elegant. We will do that in the lab. If you are really a reductionist, you can call up Rodolfo Turriaga, who may still have, from 1989, his microspectrophotometer, from which he could focus a microscope down onto a single cell and measure its absorption. He had light, and he had a detector underneath the microscope. And so here's one water sample that took him days. You put it on, you filter onto the filter, and then you focus on every single cell that was on the filter, and you get a spectrum for every cell. Personally, that seems like a lot of work. But someone had to do it. It looks great. Okay. So the absorption spectra vary a lot based on species, right? Because they have different pigments. These are just normalized. You can see big differences here, big differences here. If you look at the variance of those, you can identify different peaks of where you're seeing big differences in their pigment composition. You can assign which pigment is associated with which peak. And these are the phycobilly pigments associated with cyanobacteria. A little warning about this. These are not soluble in methanol. So if you do this approach and you have a lot of cyanobacteria, these don't get removed. You have to remove them with another buffer or water. So you just have to, if you're really interested in cyanobacteria, you have to pay attention. Okay. Ivana, oh, she's gone. I'm actually going to wait on pigment packaging to talk about that when we take a measurement. So I'm going to scooch out of there. So we see these really big differences in phytoplankton absorption in terms of whether the shape is peaked or flat. These have just all been normalized to 440. This is from a paper by Anique Burko. And we see a really big difference in the blue to red peak ratio. But you think to yourself, that's chlorophyll. Chlorophyll absorbs in the blue and the red. Why is that so different? How could this ratio change so much? Because if you have one molecule, you should have a constant ratio. You think about why you might have differences here. Other pigments. And a lot of the other pigments absorb in here. So depending on how many other pigments you have, it's jacking up the blue, right? There's also a big difference because of the size and how the pigments are packaged in that cell. And what you notice is that when you have very low chlorophyll in the ocean, so what environment is that? The oligotrophic ocean, where there's very low nutrients. The blue to red peak ratio is very high. And as you get to environments where you have lots of phytoplankton in the water, that ratio goes almost to 1. And I'm going to let you sit on that, because that's kind of a mystery. So ask yourself why that might be. Think about the phytoplankton that live in those environments, and so we'll return to that tomorrow. Okay. Phytoplankton aren't the only cells. We have protists. They tend to have this sort of exponential absorption. And sometimes you can see a big peak around 412. That's cytochrome 412. That's responsible for respiration. So if you have a nice big cytochrome peak, it means that your bacteria are healthy, or as are your ciliates and flagellates. They're a part of it, part of the community. We also have so part of the non-algal particles, they show up in non-algal particles, but they do have this peak. But that exponential slope also has global relationships that are similar, but it's much flatter. It's 0.01 compared to 0.02 for CDOM on average. So what does that tell you about non-algal particles if that spectral slope is flatter? Compositionally, what would that say about that particulate matter? If it's really steep, it's really refractory.
It absorbs more. There's less refraction.
There's less refractory material. So the particulate matter is much more labile, which means it's much yummier, which means that it hasn't been broken down as much yet. And so there's this continuum from particulate to dissolved in organic matter, but there's also a continuum in the spectral slope. And so again, the slope of the absorption can tell you a lot about the composition of the material. You could also measure every detrital particle with the microspectrophotometer if you so would like to do that. I'm going to let that one go. Okay. Inorganic particles, sediments. Everything absorbs in the blue. I'm just going to say everything absorbs in the blue. Right? Only water absorbs in the red. So you can have Saharan dust, samples from Australia. They all absorb in the blue. Some of them have some features in here having to do with iron. This is 4 different sampling locations. They have it on a log scale, so it's a line, but that means it's exponential. So basically everything absorbs exponentially. Oh, the air conditioning, thank God. Yes?
Question. So when you talk about non-alkyl particles, are you talking also about inorganic, or are you separating?
Well, ecologically, it makes a really big difference to separate them, right? Operationally, everything shows up on that filter once you've extracted the pigments out. So you can, one of the things that I've done in the past is you can then take that filter and combust it and burn off all the organic matter and then scan it again to get the inorganic contribution. The problem is that a lot of inorganic particles in the ocean get coated with bacteria. And so when they're coated with bacteria or organic molecules, you don't see the mineral underneath. And then when you burn it off, it reveals itself, but it was not there in the original spectrum. So you can't separate them by difference. So it's a tough thing. Yeah, right? So figuring out ways of doing that is really important, which I think brings me exactly, thank you. So here's an example of just such a measurement. Of sediments collected in the Gulf of Mississippi, Atchafalaya. No, it's a bayou. Yeah, the Atchafalaya, by Megastapa. It's part of her PhD thesis. And so she collected the particles, inorganic particles, and she measured their spectra. And wow, looks like CDOM, looks like non-algal particles, looks like everything I've just shown you. But then she noticed that it wasn't exactly exponential. There's tiny, tiny shoulders, and if you squint your eye, you could see there's a little peak here, maybe a little peak here. So she's like, nah, I'm going to take a derivative spectrum. So she took a derivative spectrum, and it turns out that there's very strong peaks, or not very strong, there are shoulders in here that indicate the concentration of iron in the inorganics. So you can use this type of analysis to help you guide you towards like, oh, that must have been inorganic versus organic, right? Or it's a really great way to figure out how much iron is in your sediment. Okay. And so to model the impacts of all these different things, again, the thing about absorption is that it's additive. So if I could measure all my phytoplankton absorption, and I could measure all the non-ionical particles, and I could measure the CDOM for my sample, which you will do tomorrow and the next day, you can then add the components together and retrieve, once you add water in, retrieve the total absorption spectrum. Okay. So that becomes powerful. And what it also means that in the inverse sense, if you have the total absorption spectrum, you should be able to come up with models to retrieve the components from the total absorption. And there's a bunch of different approaches for decomposing this into the components. I'm done, right as the air conditioning came on. Sorry. So tomorrow, Ivona is going to talk about phytoplankton. Yay! And then you have to hear from me one more time this week. We're going to talk about measuring absorption. We're going to do the CDOM lab tomorrow. Yay! Yay! And then Wednesday we'll do particulate absorption. And that's it for absorption. That's it. Good. Questions? How come I got the post-lunch lecture, Emmanuel? Everybody's like dying out here. Yes.
So in your early slides, you were talking about like vibration and like how the movement of the molecules. As I remember, in microwave, they're like
Rotational.
rotational. But then when you were talking about electromagnetics, what we had in like visible wavelengths, we had also the rotation.
So if you take the molecule, I'm going to best do this. So if you have a molecule and it's got an absorption at some wavelength, right? And if you had it at absolute zero, right, it would have some absorption coefficient. And then you allowed that molecule to be at room temperature. There would be some vibrational states of all of the molecules in your solution, right? And depending upon which molecules had so much vibration, vibrational energy is equivalent to an infrared wavelength, right? So if you move over an infrared wavelength equivalent in energy, it's moving over an infrared equivalent in wavelength. So that span here is equivalent to an IR wavelength in energy. Okay? And then all of the molecules also have some rotation to them. And the rotation is equivalent to a microwave equivalent of energy in wavelength. And the difference between those is microwave equivalent. So you're not in the microwave, but you have quantized energy about those electronic states. That is, the levels between those are a microwave equivalent of energy. Okay? Yeah?
So you have a slide that was talking about refractory labile particles.
Yep.
By comparison to spectral slope.
Yep.
Were those for data that had the minerals removed somehow?
So they had filtered, and they were looking at the filtrate. And so as long as there was not significant dissolved inorganic elements, like iron molecules, maybe for the Atchophyllaea, that would be tough. But certainly in the equatorial Pacific, that was organic. Yeah.
Early in the lecture, you talked about using the specific absorption coefficient and then just multiplying by the concentration to get absorption.
Yeah.
How robust has that proven to be?
So it really depends on which component and where you are. So I will say that I did a big sampling of main rivers, and we measured the absorption spectra. Were you biting me? Oh my God, it was. Got him. The absorption spectra of CDOM, right? So I had a bunch of samples and a bunch of CDOM spectra, right? And then with those same water samples, I measured DOC, dissolved organic carbon, chemically. And so I took each spectrum, each individual one, and I divided by its specific DOC concentration, right? And I ended up with a bunch of spectra that laid on top of each other, which was incredibly powerful, because then I could just go out and measure CDOM absorption and predict DOC. So I was using the inverse for this. Incredibly powerful. You cannot take this model and go to the Mississippi or the Chesapeake or the Ganges. But in Maine, for 4 different rivers, 60 different stations, monthly samples for 5 years, beautiful. Right? So yes and no. Yeah.
Why is it that this model only works in Maine? What specific component is
I think that it has a lot to do with the geologic history of the state, in that the bedrock is relatively uniform, except for over in limestone, and that when the glaciers retreated in the last ice age, they left bare rock with no sediment, and that the sediment is relatively young. So soils are very young, and the state is covered by boreal forests, and that it tends to be pretty uniform. So you could probably take this over to Norway and it would work pretty well.
I missed the question about where was it you were asking about? What state or whatever?
We were just saying, how robust are mass-specific absorption coefficients? And I said, it's very robust in Maine, but you can't take this model and take it to the Ganges. Yeah. But you can do the same thing. Get out of here. You can do the same thing with phytoplankton absorption. But again, there'll be variations if the cells are big or they're small or they're cyanobacteria or they're diatoms or they're dinoflagellates. But to first order, it's not bad. To second order, you need more information. But it's pretty powerful. We have a whole lecture on proxy, so this is a proxy. That's good. Yeah.
You showed a graph, like a 2D graph of the CDOM absorption. I didn't fully follow it.
Stop me. Everything looks the same.
Yeah, absorbing components with CDOMs is one. Is an important one.
Ah.
This here.
Yeah. Depth. Distance along the equator of the Pacific Ocean.
Okay.
This is the filtrate from a 0.2 micron filter.
Another one's both.
And the other one is, so I basically took a water sample and I measured 0.2 micron filtrate. And then I measured that same water, 0.7 micron filtrate. And if I did the difference, I got the fraction, 0.2. I think we should get some more chalk. To 0.7 micron fraction of absorption. And then I looked at the intensity of the absorption at 440. And I plotted the magnitude of that, which would be the concentration. And so you can see that this is much smaller concentration because it's a very small fraction, but it's very differently distributed. And the spectral slope of the material was very different. This had a pretty flat slope. The 0.2 had a pretty steep slope.
And then, so if you said each black dot is a
Water
point.
sample,
So
yeah.
So for example, the bottom left is the second graph. How does that shape appear?
Who knows? MATLAB interpolation. I would not. Like, don't even. Yeah.
Don't even try.
It looks like a rabbit. I do not take responsibility for MATLAB. And this is old MATLAB, too. So, yeah. Anything else? I think you guys get to go outside. No, go to the lab and play with light.
Yeah, let's take maybe 15 minutes.
So, 2:45, go to the Mitchell lab.
Go to the Mitchell lab. And so this afternoon it's a kind of free-flowing lab. You get together, 2 or 3 of you together, and there's 8 stations. The idea is that you go through and explore. There's a description that says what we might